Welcome back. So last time we talked about our neuron and neuroglia cells. This time, let's talk a bit about neuronal communication. So how do neurons communicate with one another? But before we do that, let's talk a bit about the properties of a neuron. So at its resting state, the neuron has a high concentration of potassium inside of the cell. So we have a lot of potassium, super high amounts, inside of the cell, and we have less sodium inside the cell. So not as much, right? But then on the outside of the neuron, we have a lot more sodium present and a lot less potassium present. The way that I remember this, and I will be using this kind of mnemonic, I guess, per se, throughout this, but I think of potassium as kings and the neuron itself as a castle. So the kings are most likely going to be inside of their castle. They're really not going to be roaming around. And then our sodiums are going to be our knights, per se. So we're going to have more sodium, more knights guarding the cell rather than being inside of the castle. And then later on, as we talk more about neuronal communications, obviously this concentration gradient will change. We'll talk a bit about why and how as well. But before we get there, so we have this concentration gradient, and if we were to measure the resting potential, meaning the voltage in comparison to the outside, so the voltage inside of the cell compared to the outside, when the neuron's not doing anything, we're going to have a resting membrane potential of negative 70 millivolts. Well, why is that? Why is it that we have all this positivity inside and positivity outside? Why isn't it zero, or why isn't it more positive inside? Well, we have potassium leak channels all around the neuron. It's just everywhere. So potassium tends to just want to go out all the time. These kings want to go and fight for more land. They want to go to war, basically. That's how I like to think about it. So this potassium are always rushing out, and the neuron has these channels that are always permeable to potassium, but they're not really permeable to sodium. So sodium doesn't randomly just come inside. It's only potassium that keeps wanting to leave the neuron. So because of that, we have a negative membrane potential, because obviously we have an outflux of a positive ion. So we're going to have a negative resting membrane potential. Okay, well, why is this important? Why do we need to know that the electrical membrane potential at rest is negative 70 millivolts? And once again, this is negative 70 millivolts approximately. So it depends on a neuron-to-neuron basis. It depends on organisms. It depends on a lot of different things, but it's going to be approximately negative 70 millivolts. But going back to the question as to why it matters, this resting membrane potential kind of determines what happens in terms of signaling. So let's start here. I have two neurons back to back right here, as you can see. This is our first neuron here, let's pretend, and this is our second neuron. Obviously, in order for a neuron to communicate with one another, one has to be able to receive the information, the other has to transmit the information. So let's pretend that this neuron is sending information to this neuron. And in order for the neuron to receive that information, it needs to have a structure of some sort. So that structure, once again, is our dendrite. And our dendrites are extensions of our soma. So let's pretend we have a synapse over here. That's the location where the axon terminal is really, really close to our postsynaptic dendrite. So in this case, postsynaptically, we have a dendrite. It could be a muscle cell, it could be a gland, or any other cell, but we're talking about neuron to neuron for the purposes of today. So we have a postsynaptic neuron. So our postsynaptic neuron, maybe I should, is simply just our neuron that's after the synapse. And our presynaptic neuron is right before the synapse. So this neuron, this whole entire thing, is our presynaptic neuron. And once again, our synapse is a space between the two and it's just free floating where we have chemical signaling that takes place as opposed to electrical. Because think about it, can electricity actually travel through air? Well, maybe at a really, really high amounts of it, I guess, but it doesn't traditionally travel through the air. We might see lightning, but we can't charge our devices just through thin air. So in the same way, we have something that has to happen here, right? So what happens is we have neurotransmitters that get released from the presynaptic neuron. So I'm going to try my best to draw out our terminal bouton. So let's pretend this is our terminal bouton, and we have a bunch of synaptic vesicles. These are going to have our neurotransmitters inside of our presynaptic neuron. And our postsynaptic neuron is going to have a bunch of receptors of specific type. So they don't bind to, these neurotransmitters just don't go ahead and bind to whatever they want to. They have to bind to something that accepts them. So what happens is if everything's successful and neurotransmitters are ordered to be released, this vesicle will fuse with this membrane, and eventually it'll kind of open up, and we're going to have the neurotransmitters just kind of leak out, and the vesicle is going to become a part of our presynaptic axon, terminal bouton. So let's pretend that was the case, and eventually, once again, if I were to draw this in terms of like a timeline, I guess, it's going to bind to our receptors. Let's pretend these are our receptors. And it's going to activate a series of different actions. So we have two types of main receptors. We have metabotropic and ionotropic. So what's the difference between the two? Metabotropic kind of has another protein that's associated with it. It's called a G protein that is connected to the receptor. So let's pretend this is our receptor per se, like a V-shape-ish, and then we have our G protein stuff here. We have our different subunits, your alpha, beta subunits, which kind of interact, and later on a chain of reactions happens inside of the cell telling the receptor, hey, maybe we can open up and allow for, sodium to come through, chlorine to come through, or potassium to come through. It just depends on what type of receptor it is. Now, ionotropic, on the other hand, is just a simply ligand-gated neurotransmitter receiving channel. So we're just going to have, let's pretend this is our channel. This is much more blown up. And obviously we're going to have some sort of blockage here, right? These two are pretty close to each other. When a neurotransmitter comes and actually binds to the site, it says, hey, okay, let's open up and allow for whatever ion to come through. So then the ions will start flowing in, and that is an ionotropic. Obviously, taking a look at the two scenarios, right, where we have a G protein coupled receptor versus a ligand-gated receptor. And once again, these are the same thing. So if you ever hear G protein coupled receptor in association with neurotransmitter receptors, it's the same thing as metabotropic, and ligand-gated receptor channels are always going to be ionotropic. So why is that important? Well, metabotropic, right, obviously has a series of reactions that happen in order for the receptor to open. It's going to be much, much, much more slower, right? Ionotropic, simple, just attaches and it says, hey, let's open up our receptor. Let's allow the ions to flow through. So we have sodium coming in or potassium coming in. It's at a much more faster rate, just like an on-off switch, versus where you turn on something and then you wait for another thing to turn on and then go through and activate the rest of it. So kind of think about it as a chain of reactions for metabotropic, and for ionotropic, it's a simple on-off switch. Great. So now we have all of this, these neurotransmitters are coming in, they're binding to their specific receptor. Now what? Well, why is that important? Now, if you think about it, this isn't just the only presynaptic neuron that's attached to this postsynaptic neuron. Our neurons are very, very complex. Our neuronal networks are also super complex. So we have a lot of different things that are connected to one another, different types of synapses, so on and so forth. And so in this case, right, you might have another neuron that's, attached to this thing here, another one attached to here, whatever, right? It's just all over the place. And one neuron is going to have to take in that information, and poor thing will have to sort out what to do with it, right? So there comes, or in that point, we need something to summate all of those signals. Say, hey, actually fire or don't fire, right? Because we're not actually at the point where our neuron is firing. We're only at the point where it's receiving that information. So we have a bunch of different dendrites, right? Dendritic spines as well, that are connected with our synapse. And that synapse takes in so much, different neurotransmitters each time and each different synapse. But now, let's say we have one here, another one like right here, right? Those are two different synapses. One of them might say, hey, here's a neurotransmitter, bind and open up a sodium channel. The other one says, hey, open up a chlorine channel as opposed to sodium or potassium. Well, now we're kind of, in a troubled spot, right? Well, what happens then? They kind of cancel, right? This is where EPSPs and IPSPs come in. So EPSPs stand for excitatory postsynaptic potential. This is when a neurotransmitter binds to a receptor, right? And I'll use a ligand-gated for this example just to make it simple for us. And it says, okay, well, it binds and it opens up this channel. Most of the times we're going to have sodium come in. And then in the case of IPSPs, we're going to have chlorine come in. So this is just another neurotransmitter, just changing colors so that you can kind of see it. Once again, I'm going to use a ligand-gated. You can have a G protein coupled receptor as well. And this will cause chlorine to rush in. So by nature, chlorine is negative, right? That's molecule. So if you think about it, if there's enough sodium and chlorine to come together, they'll just cancel out. Or if there's more sodium, then, well, great, we have a lot more positivity. If there's less sodium, then it's not going to be as positive, right? Or if there's more chlorine, same thing, right? It's going to be more negative. It's not going to actually fire. So this brings the concept of temporal summation and spatial summation. So temporal summation, if you think about it, is just kind of like over time, right? Temporal, time, very similar. So if you have a bunch of EPSPs, they all are back to back to back, right? Well, it's going to be an overall positive. They add up over time. Think about it this way. You're in your car, and you have your accelerator and your brake pedal, right? If you keep pumping your accelerator, right, just pump it once, pump it twice, you're eventually going to speed up, right? If you pump it fast enough over a period of time. So they all add up, right? Same thing here. Your EPSPs add up to a positive overall charge inside. So your resting membrane potential is increased, and they're added up. So chlorine, I guess, with your IPSPs, think about it as your brake, right? So if you had an IPSP followed by EPSP, obviously one IPSP, two EPSPs, it's going to be an overall positive. It also depends on, the how many IPSPs, how strong it is, I guess, in terms of how many synapses fired an IPSP or gave us an IPSP. So that's going to be overall positive. That's temporal summation. So over time. What about spatial summation? So similar to how it sounds, spatial is over space, right? So if you take multiple different locations, right? Let's go back to what we were talking about here. We had this synapse here. We had another synapse over here. Let's say we had another synapse over there. Let's pretend, right? And if all of them fire an EPSP, then all of them, eventually get added up right around somewhere in the soma, and the integration finally finishes somewhere here, known as the axon hillock. also known as your integration zone, right? So everything kind of summates there, and it decides what is going to happen. So for this example, think about it in terms of maybe a pot of water, right? If you add water into a pot from different locations, right? Let's say like there were multiple pipes or something, and if you add water from all of those pipes at the same time, well, obviously you're going to fill the pot, right? Same way, spatial summation has to do with multiple different locations at the same time, so they'll eventually add up together. And now that we understand that, let's talk a bit about the electrical side of things. So great, we have our EPSPs and IPSPs. What are those, by the way? They're known as graded potentials. Why? Because you can kind of change them slowly, right? It doesn't suddenly change everything at once. You can have an EPSP and an IPSP, they add up, and it'll change the resting membrane potential a little bit. It doesn't actually cause it to fire immediately. Well, with the electrical side of things, that's going to be our actual action potential, like our electrical part of it. I'm going to call that AP for now. So that action potential is a fire or don't fire type thing, you know, and that happens at a membrane potential of negative 55 millivolts. Once again, approximately. This is known as our threshold potential. So at this threshold, it fires and things go on. Okay, let's think about it in terms of a graph as well. I want to kind of show you how it would look on a graph. You might see this. So on a graph, right, let's pretend this is 0, and everything is in terms of millivolts. Let's pretend this is negative 55, and then negative 70. I should probably increase my axes a little bit. And then we have our positive side. I will just arbitrarily call this positive 40. Okay, so initially we have our resting membrane potential, and this is time, by the way. This is just over time. We have our resting membrane potential. Nothing's really happening. And in order for an action potential to occur, we need enough positivity to raise the resting membrane potential from a resting state to our threshold of negative 55. So we might have an IPSP, which brings it down, an EPSP, and then an IPSP, and eventually it'll hit negative 55 if it wants to fire. What happens if it just stays under negative 70 or at negative 70? Well, nothing happens. So that's important for inhibition, which we might talk about later. But once we hit negative 55, well, now things start to change. So going back to our neuron, there are a bunch of voltage-gated channels across the axon. So those voltage-gated channels, they'll start to open up. They're like, hey, let's start, opening up, especially because our threshold is reached. We're going to allow sodium to come in. So those knights are going to retreat back home really, really, really quickly. They'll come right back in. They'll rush in. So obviously that's going to increase our membrane potential, right? So with that increase of membrane potential, it causes a series of things to happen, and eventually we will have a firing where it reaches our neurotransmitters, and we have a release of neurotransmitters. We'll talk a bit more about action potentials soon.